I'll be making my presentation in Russian with the slides in English. As Natalia has mentioned, I'll be talking about the molecular organization of the human brain. There are various approaches to studying the brain. You can study the behavior. On a deeper level, you can study the electrophysiology and connectivity, neuron activity, and the way the neurons connect with each other in the human brain, the way they are arranged in neural networks, and the most profound level, the deepest level, is the molecular biology, the area we are involved in here in Skoltech. And I would like to share a few examples of the research being done in Skoltech and the outlook for human brain research at the molecular level. The human brain is a very complex structure with a very complex anatomy and very complex structure. And when we analyze various cell types arranged in various anatomical structures, we observe different molecular components. First of all, it's water, but we don't research water in Skoltech now. RNA, those molecules which transfer information from DNA to proteins, and polar metabolites, which are the basic molecules which store energy, and lipids, which are also energy molecules, neurotransmitters, and are involved in transferring other types of signals. When we analyze this data, we try to reconstruct brain structure following those changes that we observe at the molecular level. As Natalia has mentioned, we observed that over time, because when a person is born, brain is developed only by 25%, and you have seen the way it develops over time. But the most important thing about the human brain, about the cortex, is that the neural networks for the newborn, they're not there yet. And if we look at the way neurons connect, and the neurons, they're responsible for controlling our behavior, you see that those connections, they are just emerging in newborns, in children. And when we look at neurons and synapses, so here you see a picture of a neuron with lots of dots. Those dots are synapses, synaptic contacts that neurons use to communicate with each other. But this is a neuron of an adult. If you look at a neuron of a newborn, of a child, it will be just a neuron without any synaptic contacts that an adult has. Well, the thing is that the human brain develops under the influence of the environment. And we don't know how we will respond to the environment in the future, and how we will structure our communication with other people, about how we will respond to dangerous factors, for example. So these all influences the human brain development, all the processes behind the human brain development, and synapses are just one part of this overall development picture. Let us see whether we will be able to reconstruct the brain structure by analyzing the RNA activity and proteins. So in this picture, you see that DNA is present in all the cells. DNA is basically an archive. a library of all the genes which govern the way our body develops, not only the brain as such, but other organs as well. The DNA is pretty much the same across all the body organs, with some minor differences, but they're not significant for the purpose of this talk. It's just that in liver, for example, one group of genes is triggered in another organ, a different group of genes is triggered, and those gene combinations, they change across the body. If I look at the amount of RNA, which is being read from the genome, we can deduce the kind of genome which was active at this or that period of time. If we look at the evolution of the cognitive function, we may assume that there must be some special molecular substrates in the brain. And we could compare our brain with the brain of a chimpanzee. This is what we did in our lab. We have a huge collection of brains. This is the frontal lobe, the frontal cortex, which is responsible for our behavior and interaction with the environment. There's a similar set of samples taken from chimpanzees and macaques. We have identified a group of genes responsible for the peculiar development of the human brain, different from other species. But what are those genes? Let us try and reconstruct what they are responsible for. It turns out that the genes are responsible for building the synaptic contacts, those knots responsible for transmitting signals between the neurons. Because neurons, well, they can't really talk to each other. They only have an electrochemical communication with each other. So one neuron triggers another neuron, or one group of neurons triggers another group of neurons, which in the end ultimately govern our behavior. This is what we can see through an electron microscopy. You know, in biology, we have to believe only what we can see with our own eyes. Because if in mathematics everything is based on abstract concepts, in biology you only believe in what you see with your own eyes. Otherwise, no one would believe you. So here, the electron microscopy helps us to detect those electrical synaptical contacts, and we have been able to count the number of synaptical contacts in the frontal cortex of a human and of a macaque. And they're shown here in this diagram. So when a human baby or a macaque is born, there are no synapses whatsoever. Because, well, you know, the frontal cortex is responsible for interacting with the environment, and the newborn, it doesn't have any interaction with the environment yet. Of course, some basic instincts, they are there, but some complex social behavior, some complex social interaction with the environment, it develops over time only. That is why it is so important to pay attention to the proper social development of a child. The number of synapses peaks by the 5th year of life, and after that the number goes down. Why? Well, because our behavior patterns become a routine for us. So by the age of 5, a kid knows how to interact with the environment, and he needs less new synaptic contacts, less synapses. Because when you go shopping, for example, you usually shop for the same things. It's a routine, it's a pattern. Otherwise, we would spend a whole day shopping, not knowing what to buy. In case of macaques, the number of synapses, the number of new synapses peaks by the age of a few months. So by that age, a baby macaque knows how to interact with the environment, and after that, it all becomes a routine. And the number of new synapses goes down. So we have a very different dynamics here, a very different trend. Chimpanzees, they're much closer to humans than macaques. We were the same species just some, I don't know, 7 million years ago. But the qualitative shift that we have observed during the gene expression stage, we see that the number of new synapses obeys the same dynamic across humans as across chimpanzees. So we have identified certain changes at the gene level, and then we reconstructed the brain structure, one of the processes in one of the brain regions. Again, I have to remind you that the human brain is a very complex structure. Before we have analyzed only one process, only in the frontal cortex. So some people think that human brain is just really enlarged ape brain or a bad brain, but it's not true. So we have to analyze the human brain at a deeper level in order to be able to identify the unique features of the human brain, which differ from other species. Another part of our lab's research is dedicated to studying lipids, fat cells. Well, some people may say that there's nothing unique about the brain fat composition, but you know, the fat cells, they're used in cell membranes, and this is where all the interaction happens between all the receptors, between all the cells, and the properties of a membrane do affect the property of this communication between the cells. Well, we know that, and we know that if you try to dry out the brain, dehydrate it, you will be left with the fat cells. But how does that affect the brain functions? It's all about the emergent field. Well, the scientists started the research of lipids some 60 years ago, but there have been significant breakthroughs and advances over the past decades. And lipids do represent the physiological condition of the brain over time. We have all the technologies for studying that, like mass spectrometry, which enable us to collect data on hundreds and thousands of lipid compounds in the brain. Then we decided to take it further, to analyze what happens in a healthy brain compared with the processes we observe in a damaged brain of a person diagnosed with schizophrenia. I know that the brain has a very complex anatomical structure with different responses in different regions. So we took various regions for our analysis, like primary cortex, secondary cortex, and you name it. We compared lots of different groups And here we analyzed the several brains of schizophrenics and 4 brains of healthy brains. Well, the bad thing about this experiment is that we were studying the brain of a dead person, of dead people, but you know, the lipids are still there. When they discovered the city of Pompeii, which was buried under volcano ash, all the things remained intact. And by analyzing the remnants of that city of Pompeii, the scientists were able to reconstruct the life of the citizens before the tragedy. We're doing quite the same thing. So we take a dead brain, but in terms of lipids, all the lipids are there, intact, all the buildings are there, all the plates and cups are there, and we can reconstruct what was going on there. This project is still ongoing, so I cannot share the final conclusions with you yet. But those blue dots, these are samples of 75 regions of the brain with 4 control regions, which don't have any cognitive pathologies, and the orange dots, these are the samples taken from the brain of a schizophrenic. And we see that there's quite a lot of difference in one case and less difference in another case. And this is the coloring of those samples by anatomy. We will see it now in more details, and we see that there are lipids which are typical for the myelin, lipids typical for the gray matter, for neurons, they are shown in yellow. There are lipids responsible for the emotional roots, those greenish. And if we look at all the 175 regions, well, you won't see the names of those regions because they're in a very tiny font here, but the size of the bar shows the amount of change in lipid composition, the size of variation in lipid composition between the brain of a healthy human and the brain of a schizophrenic. If the bar goes down, the number of lipids goes up, and if the bar goes down, then the number of lipids goes down. So we have a number of clearly defined aspects here. There are certain regions where there is no difference whatsoever between a healthy human and a schizophrenic. The second aspect is that usually the changes go either up or down, so the number of lipids either increases or decreases. And we see that the green color is dominating here. The green color shows the myelin. So the number of lipids in the myelin either increases or decreases. And so the connections between lipids represented by myelinized axons, it either increases or decreases. If we look at the regions where the number of lipids goes up or down, we see that the main decrease in the number of lipids is observed in the white matter and the midbrain. The white matter is where we have the so-called power grid in our brain, and the schizophrenics lose this connectivity between neurons. So there's much less connectivity between the neurons and less connectivity in the cortex. There's more white matter, there's more lipids in the associative cortex, in the basal ganglia, and in limbic cortex. So this is a pathological growth of such connections in different regions of the human brain diagnosed with schizophrenia. So we see how it works here on the brain map. The white matter grows in some areas and it shrinks in other areas. But what about the practical meaning of that? Because you can't really take a live human and start cutting into the brain. Well, we try to identify those differences in blood plasma. So for that, we carried out quite a large-scale research in cooperation with the clinical institutions of 3 countries. We collected the blood plasma of people diagnosed with schizophrenia in Germany, Russia, and China. We analyzed that on using mass spectrometry, and here we show different lipid classes by different colors. And here we show the same colors in the form of bars. So we see how the number of lipids in different classes either goes up or down. And we see that in all the 3 cohorts, German, Chinese, and Russian, we have uniform changes in different classes. So here we observe a very strong correlation. So in this slide, we show the data set for carnitine with a lipid tail, 18 carbon atoms and 2 double links. And here we see that the concentration of carnitine in the blood plasma of the control group and of the people diagnosed with schizophrenia, well, of course, there are some outliers, but if we were to draw a border here between the 2, by the amount of carnitine in the blood plasma, well, here we would see that the concentration of carnitine goes below 20. Of course, this is not a direct indicator of schizophrenia, but this is quite a good proxy marker. Some of the changes that we detect in blood plasma, they have inverse correlation with blood plasma. So if something shrinks in the brain, it increases in the blood plasma. At this point, I would like to finish my presentation and let me say a few acknowledgements to all my colleagues who participated in this research, and thank you for your attention.